My name is Greg Albery and I'm a postdoc at Georgetown University. I'm going to be talking today about how age changes social behaviour and the possible influence on infection dynamics in a wild deer. So an individual's infection status is fundamentally determined by its exposure to parasites and its susceptibility to those parasites when exposed. More contacts often results in more exposure events, and therefore social behaviour is often an inherent determinant of an individual's exposure rate and therefore of its infection status. There are lots of reasons to expect age-related changes in sociality and therefore infection, but we so far don't know the answer to this question. How does social behaviour change with age, and what are the potential consequences for infection? Ecologists working in wild populations increasingly appreciate that older animals senesce in terms of fitness, behaviour, and these are both shown in this one population of deer that I'm going to be talking about. Animals often also senesce in terms of their immune systems in a process called immunosenescence, and this was shown in the soy sheep. And there's some emerging evidence for declines in social behaviour, as in this example in chimpanzees, and it's possible that that could alter exposure to parasites. So the question I really want to ask is, how do age-related changes in sociality and therefore exposure correlate with age-related changes in immunity or susceptibility? And could a negative correlation between those two things be a sort of adaptive response? I'm asking this question with the Isle of Rum red deer, which is an island off the west coast of Scotland. It's home to 300 plus individuals, which has been studied since the 1970s, and they're censused 40 times a year with really detailed behavioural and life history information. I have individually tied helminth egg counts, which I've been collecting since 2016 during my PhD, and that's given us really fine resolution infection data. Parasites correlate with season, spatial positioning, reproduction, and fitness. These are my collaborators. It's my supervisor and co-supervisor for my PhD. It's Josephine and Dan, and Josh Firth at the University of Oxford, and Tim Clutton-Brock at Cambridge. So together we did these analyses of social behaviour to find why social senescence might be happening. We used the integrated nested Laplace approximation, GLMMs, with about 6,000 annual behaviour metrics, taking averages for 900 or so females. I don't have time to go into the inner workings of INLA, but if you want to know more about how it's used to control for spatial autocorrelation, then shoot me a message. With that, we ask, does social connectedness change over the course of a lifespan? And does this occur through either selective disappearance of certain individuals, demographic changes in spatial behaviour like habitat selection, or within individual declines in social behaviour? We found that older individuals do become less social, so the age on the x-axis and smaller group sizes on the y-axis. They also have fewer unique contacts and weaker connections to those contacts. These findings are robust to substantial spatial structuring, and there was no evidence of selective disappearance or of demographic structuring effects. So this shows that there's a within individual decline in social behaviour or social connectedness that's happening. So what effects could this have on parasites? We use these non-invasive fecal egg counts that I collected by following individuals around with a telescope and waiting until they poo and then collecting it. Inside the poo, I found these strongyles, gastrointestinal nematodes, and worked out whether the parasites are being driven by social behaviour, spatial behaviour, age, or a suite of other drivers like the ones that I mentioned earlier. We found that both age and sociality influence parasite count. Individuals who are better connected, so you see degree centrality on the x-axis, have more parasites on the y-axis. Older deer have higher parasite counts as well. But individuals who age and decrease their social connectedness as they age end up with lower parasite counts. And there were no effective spatial behaviours, interestingly. So putting this together, we find that age has contrasting effects on infection. Older individuals have lower sociality, but because sociality has a positive effect on parasites, age has an indirect negative effect on infection, which contrasts with age's positive effect on infection, which was a more direct effect. So in conclusion, we find that aging deer are less socially connected, partly due to altered spatial behaviour. Less connected deer have fewer parasites, not due to altered spatial behaviour. And age-related reductions in connectedness result in lower infection, which contrasts with direct age-related effects. Parasite avoidance could therefore be driving social senescence, because older individuals might be trying to reduce their sociality so that they can reduce their exposure to parasites, potentially to counteract their waning immune systems. To find this out, up next, we have to include how do changes in immunity fit in with the whole picture. And I personally want to find out why there were no spatial behaviour effects as well, because I find that quite intriguing. We need to do a load more models, but I think this is a kind of exciting finding. Thanks for listening.